All right, we're going to get started here in a minute. Excuse me. So I uploaded the slides ahead of time this time. I'm going to try to keep up with that habit so that if you guys wanted to follow along or make notes as we're doing this, those are available to you in that discussion for week 2. So you go to discussions and you go to weekly reviews and find where I post about week 2. Right now, it's just the PowerPoint slides. And then once the video is uploaded, I'll post a link to that. And AI for Zoom automatically makes a summary of the meeting. And so I'll post that too, so that when you guys are studying, it does kind of go over the major topics and stuff that we talk about. I don't ever edit those AI summaries. So if they have something weird or off about it, that's because the AI put that there. And so just FYI, but take it with a grain of salt. They're actually pretty good at summarizing what we talk about. So I found those to be pretty useful, especially for those of you who are just looking for, kind of summaries to refresh your memory or whatever. So let's get into what we're talking about today. Chapter 4, we mentioned bacteria and archaeas. We'll go over that first as our major topics for chapter 4. Why is this not? Yeah, okay, there we go. So we'll talk about our major covered topics for that. Basically, highlights that we went over in the lecture, and then our most missed topics, and this is in the past on exams. I do focus most on the most missed topics for obviously the most commonly missed topics, but then I also go back and make sure I hit the most commonly missed topics specifically for my hybrid section, because they tend not to do as well as my traditional sections, mostly because the lecture portion really is relying pretty heavily on you guys, doing the work for yourselves. And it tends to be that people that tend to enroll in the hybrid section, not everybody, but it tends to be the majority of my students that enroll in the hybrid section during the traditional semester, not the summer, but just the traditional semester, that those tend to be the students that expect to be doing less work. And this is not my class that I'm, assuming this was from all campus wide. So that has been something that we have been discussing as a campus to try to, if you're more likely to enroll in hybrid because you think it's going to be less work and then it ends up being more work. And then that could be why those students are on average doing worse. Like I said, it's not my class. And if you guys have taken a hybrid course and they didn't warn you about that and you were struggling with it, that's their fault for not warning you about that. But it tends to be a trend. And so we're trying to work on how we can increase, improve the experience for our hybrid students during that traditional semesters. During the summer semester, it tends to be that the hybrid students that are taking classes in the summer are more motivated to begin with. But if they're taking a hybrid class in the summer, they're typically are thinking about the fact that they're going to have to be more self-motivated, and they do usually as well or better than my traditional classes in the regular semester, 16-week semester. So also summer class is way more intensive. You guys think that, we meet a lot and spend a lot of time in class and lab so far. We haven't even gotten to the point where we're meeting twice a week, really, with lecture and lab, and you'll get that going next week. But it's this summer, we meet Monday, Tuesday, Wednesday, Thursday in lab, and the lecture is entirely online. So we have lab every single day. And so that keeps it going. Like, you don't get a break in between. And so that means you have to keep up with the work, but you're also not forgetting about what we've learned the day before and all of that. So I don't know. It's really hard on me teaching those classes. I can't teach more than 2 sections at a time because of that. But summer class is way more intensive. I think that's part of why the students do better is because it is more demanding, so they have to step up. Um, yeah, so that's why we're doing this. Is if you are hybrid, I don't want you guys falling behind. I want to keep you guys aware of some of these harder topics that get sort of a little lower grade from a lot of people. So, chapter 4, bacteria and archaea. Here are our key concepts for chapter 4. We have our bacterial cell structures, shapes, and arrangements. So we have a lot of the main structures that we covered kind of listed out here for us in this, but our shapes and arrangements, I do want to just briefly touch on those. The shapes of the cells, we're talking about bacteria here. Not that archaea is different, it isn't, but I had a student bring up about, shapes of yeast. So yeast have this shape and the yeast do this and that. And you could potentially say, if you're looking at individual yeast buds, they have sort of a cockeye shape to the buds, but the way that they branch off from one another, they're sort of pleomorphic. That's because yeast are fungi. They are not bacteria. They're eukaryotes. And so that's why I want to focus a little bit on we're talking about bacteria. This is the bacteria and archaea chapter. So these are traits about bacteria and archaea. We'll talk about traits of eukaryotes next week. Okay, so these are akaryotes. So don't forget that. So what's the defining feature of akaryotes versus eukaryotes? It is in the name. You just have to know what the breakdown of the words is. So the akaryotes, they don't have a nucleus. And here, when we're talking about a nucleus, we're talking about that membrane-bound organelle that contains the genetic information. In fact, akaryotes don't have any membrane-bound organelles at all. So the nucleus might be like your base, like your starting point there, and then past that you would have more membrane-bound organelles. But these guys don't have any membrane-bound organelles. They don't even have a membrane-bound nucleus. So that's their defining characteristic. So that's what'll be, if you don't have a nucleus, you're falling into the akaryotes. The two groups of akaryotes are bacteria and archaea. So now talking about the shapes of bacterial cells, those shapes can be cocci or they can be bacilli. And yes, we did talk about curved and spiral and all that, but these are the two biggies that you for sure are going to be tested over regularly throughout the semester. So definitely these should be your focus. Cocci, these are our round or spherical ones. And the bacilli, these are rods. So our cocci really are going to have a nice round shape to them typically. And the rods, they're going to be sort of like long pill shaped guys. It looks like rods. And the way I remember is that these letters here in cocci, they are very round shaped. So cocci, those are the round. And then the bacilli, they've got these two L's in here, and that those L's look like rods to me. And that's how one way you can remember which one means which. And those really are the biggies. In the lab, so in the lecture portion, the lecture tests, the lecture exams, when I'm talking about cocci and bacilli, you're going to just need to know what the terms mean. Definition wise, you should just have that simple understanding of, if I'm talking about cocci, you're talking about round cells. If I'm talking about bacilli, I'm talking about rods. And we're going to talk regularly about organisms where I'll be like, well, these are cocci, so blah, 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 or these are bacilli, blah, blah, blah. I'm not going to reiterate every time necessarily that they're going to be round versus rods. Same thing with gram positive and gram negative. As introducing these topics in this chapter, when we move forward into units 2, 3, and 4, I am going to be talking very regularly about gram positive versus gram negative, and I'm not always going to be reiterating what that means. It's very important that we're picking up the foundations in unit 1, because we're going to be using unit 1 information all of the time in the rest of the semester. So it's really important we're getting those basics down. So anyways, our shapes of our bacterial cells, our cocci and our bacilli, those are our biggies there. And then our arrangements. This is basically just how those cell shapes can exist arranged. Typically, when we're looking at them in our microscopes as slides, fixed slides. But this is how they would live in arrangements in the environment as well. So the main arrangements that we talked about was Staphylo. So this means irregular clusters. I wish I was writing this smaller. All right, and then Strepto. These are strips or chains of cells. All right, so that's some biggie groupings. Now, I'm not going to write on here diplo. I mean, come on, guys. If you guys don't know what diplo would stand for, it's talking about pairs, right? So that's pairs. So staphylo, irregular bunching, strepto, strips or chains, and they're literally going to be in long chains, and they'll be very, very visually apparent that that's happening. And then we have palisades. And so palisades is like when the rods are standing up next to each other in a fence post arrangement. So palisades, we really only ever see this in the bacilli. I mean, if you were to take cockeye, you were to take cockeye and stack them up next to each other, there's really no separating whether that's vertical or horizontally stacked. But the bacilli, you could arrange them end to end like this. This would be strepto arrangement for both of these guys. Okay. But for the palisades, for the bacilli, this is going to look like this. Okay. So that's now we're talking about vertical stacking like a fence as opposed to end attachment, which we see with strepto. Staphylo, the irregular clusters, is typically something we really only see with cockeye. And this is just because it's just the way that the cells are kind of splitting off from one another. And if we were talking about staphylo arrangement with bacilli, it just looks more random and it's hard to distinguish from just randomness. I want to be very, very clear here when we talk about cellular arrangements of our bacteria. You can have your cells organized in arrangements, but a lot of times they're just randomly existing. They're not necessarily staying in an arrangement. But when cells exist in arrangements, it's usually quite defined and falls within these defined categories. Okay. So obviously, if we're talking about Staphylococcus aureus, it's the name of that species, staph that we talk about with MRSA. Staphylococcus aureus tells you in its name that it has a staphylo arrangement. So we've got irregular clusters and it's staphylococcus, so you actually know that the cell shape would be cockeye or round. So it's telling you have irregularly clustered or grape-like clusters of round cells. And that's going to be a rule for staphylococcus. So I just wanted to touch on that because we didn't have much info on that on here on our little key concepts thing. So moving on, capsules. These are sugary coatings. Wow, that was really a lot thicker than I thought it was going to be. These are sugary coatings that bacteria will use to adhere to surfaces. They'll use them to evade our immune system. The term that we introduced regarding that was phagocytosis. So that would be whenever our white blood cells are eating up, phago means eat, and they are cells, so cyto, and the cells are eating up like foreign material or invaders. So that phagocytosis process, that is going to be inhibited because they can't grab on to the bacteria as easily because they have this sticky coating on them, the capsule. It's also helpful for forming biofilms. Flagella, these are appendages on bacteria that they use for motility. And this is the only appendage that bacteria will have that is going to be relating. I don't know why these are so thick. That's going to be relating to that motility. Just get rid of some of this really quick so we can make some space if we need to write. So if you have bacteria, and not all bacteria are motile, not all bacteria can move around in their environment. So those that do move around in their environment are capable of moving around in their environment. Those guys are going to have flagella always. So flagella are for motility. It's just a little thinner. Let me go ahead and make this thinner. Flagella. There we go. That's better. They're for motility, for moving around. If you are modal, you have flagella, at least one. There's all different arrangements that you can have of those flagella, and the terms for those were leading with basically something prefacing this word, trichus. And this trick part, that means hair. Okay, so it's describing like hair-like appendages on these cells. If you have just one, that's monotrichous. If you have it at each end, both ends, let me actually put both so that we're very, very clear about this. Both ends. So we're talking about rod shapes here, right? Now, if you were a round shaped thing, you don't have ends, right? So that's a little harder to find. But if we're talking about rod shaped guys, now we have ends. And if you have flagella at both ends, like that, then you are amphitrichous. So remember, amphi means both. If you have flagella in tufts at the end, one end or the other, so we have groupings of flagella. This is called lophotrichous. And then if you have flagella like all over, this is called peritrichous. So that would literally be just covered in these flagella. Okay. So that's the arrangements we can have if you have flagella and you are modal. All right, so our cell envelope. The cell envelope is literally the entire outer protective and controlling surface of these cells. So everything is on the outside. So you have all your nice goo that's on the inside that contains your DNA and your proteins and your sugars, your ribosomes, everything that is going on in the cell. And then you have everything that's containing all that. And so always your cell envelope is going to consist of the inner membrane, which we call the inner membrane is the cytoplasmic. All right. Most bacterial cells also have a cell wall. Let me make it purple actually, because, all right, so we're going to do the cell wall. And so I'm going to put it like this, just so you know there's some thickness to it sometimes. So this purple part would be the wall. And then some bacteria. Oh, that's not what I meant to do. Oh, there it is. Draw. It's like, where did it go? Or some bacteria will have an outer membrane. Okay, whatever. An outer membrane, and we really only see this in gram-negative bacteria. Okay. All right, so that's the whole thing. This is the cell envelope. That entire thing all together, this whole mess, all of these features, whatever you have going on your outside combined, that's your cell envelope. Okay, so the cell envelope encompasses your cell wall and whatever membranes you might have for a bacteria, and that's important. All right, so that's the cell envelope. For gram-positive bacteria, their cell wall, let's clear this out a little bit. For gram-positive bacteria, their cell wall is very thick. So I'll just put G plus, thick wall. And then for our gram negatives, they have a thin wall plus outer membrane. Okay, so that's the difference between gram positive and gram negative. literally like that. This is our defining difference. And what you guys need to remember, and I'll be drilling this into your head so freaking much over the course of this semester that this is going to become very commonplace. But I do want you guys to know, like, we have the unit 1 exam coming up, basically after week 4. So week 3, we are going to, on Monday or Tuesday, depending on your section, on Monday and Tuesday, we're going to be lecturing over chapter 5. On Wednesday and Thursday, chapter 7, right? And then that's week 3. Week 4, Monday and Tuesday, I'll be doing the review, and it's me giving my mini presentations over the major topics in Unit 1 for examples for you guys to follow from for your own presentations when you get to yours in Units 2, 3, and 4. That's what we'll be spending that whole day on. And then on Wednesday and Thursday of week 4 would be our exam. So you got 2 weeks till our Unit 1 exam. And so quite literally, a day or two shy of 2 weeks. So that's coming. And it's going to be like that for the rest of the semester. Pretty much every 4 weeks, you're having an exam. It's pretty common for most classes, just we don't really necessarily think about it in that context. And it seems like it goes by pretty slow, but we have only had we've had 3 lectures. So, so we've only got 3 more lectures, a review, and then the exam. So keep that in mind. That's pretty much how this course is going to be trucking along. So learning the gram positive and the gram negative, you got that time to prepare. You've only got that time to prepare for your exam. So if you haven't actually started studying and reformatting your notes, you're behind. Okay, I'm telling you, you are. And you probably are. You're just in denial about it. Okay, so that's our gram positive and our gram negative, and the difference between those. Our cell wall, that wall, whether it's thick or thin, if you have a cell wall and you're a bacteria, your cell wall is made up of something called peptidoglycan. And we talked about how that term tells you what that really is. Peptidoglycan is a scaffolding, basically, of chains of sugars. So we'll have many chains of repeating sugar units. Okay, so that's what the orange will represent here. So chains of repeating sugar units, and they are cross-linked to one another, like kind of alternatively, by these peptide chains. And what a peptide is, it's a short sequence of less than 20 amino acids linked together, just acting like a little bridge between those sugar chains. So peptido is that peptide cross-linkage, and glycan is talking about those chains of sugars. N-acetylglucosamine and N-acetylmuramic acid, which I will not test you on those, but that's what they are. So we get a sense of kind of chemically what's going on. And that's the cell wall for bacteria. The only deviance that we have from this would be bacteria that have mycolic acid. So instead of having just a thick old cell wall of just peptidoglycan, what these guys have down here with mycolic acid, they have some peptidoglycan, but instead of just peptidoglycan, they will also have these like wax. That's not really a very dark color, is it? Let's do something else. Because they have this wax that's kind of attached to that at the top. So this makes them pretty resistant to their environment. This makes them, able to be resistant to chemical cleaners and not be as likely to dry out, and it's easier for them to survive in what would be otherwise a drying environment. And these guys live in areas like on your skin or in your lungs, or areas that could be potentially dry. So they're less likely to be dried out. And those lipids, remember, waxes are lipids, so they're hydrophobic. Told you I was going to throw these words around a lot. All right, so hydrophobic lipids that are going to be water repellent, but they also repel the stains that we typically use to stain cells. And they might react weird with the stains anyways, unpredictable. So we can use a special stain that reacts with, and we call it an acid fast stain. So when we stain these cells with a lipid soluble stain, so it prefers associating with lipids, not charged things, not polar things. So associate with the lipid, which is our mycolic acid wax. And so when we rinse this with our acid, which is notoriously charged, acids are going to have extra um, H plus, protons, hydrogen ions. So more charges there. This doesn't care about a charge trying to rinse it away because it's lipid soluble. So it doesn't care about some charges coming along. Whereas our other stains that we use to stain cells with, they are going to have charges to them. And so acid would affect cells that have charges and are normally associated with stains with charges. So we rinse it with an acid alcohol. Starting to sound familiar why we have acid alcohol in your stain drawer now. The acid alcohol that we use will wash that stain away from our charged cells, but it'll stay associated with cells that have the wax on them. This is always going to be certain species of bacteria, Nocardia and Mycobacterium. The only one you guys have to worry about is Mycobacterium. So these guys will stain gram positive, but if we were to do an acid fast stain, they would be acid fast positive as well. And so we'll talk about that in exercise 4 in the lab. We'll actually get to do that. But the exam is actually on the day that we do exercise 4. I'm pretty sure. Yes, it is. So spoiler alert, you're going to be doing exercise 4 after you've taken the exam for most of you guys. So you won't get to do this exercise before the exam and probe for the understanding what's a hands-on thing. This happens after. So just heads up, that's going to be going down like that. So there's that. And then these are always Mycobacterium. So they're literally the name of the bacteria that have these will be Mycobacterium something. So we're talking about Mycobacterium tuberculosis, Mycobacterium leprae, Mycobacterium avium, which is Mycobacterium avium causes tuberculosis. It's similar. It's the same kind of disease as Mycobacterium tuberculosis proper causes, but it's like only going to be seen in AIDS patients typically. So that lowered immune system makes it so the avium complex bacteria, those guys will cause the same symptoms as regular tuberculosis would, but in immunocompromised folks. So that's still medically relevant because I hate to break it to you, a lot more people have AIDS than you think. A lot more people do. And we're talking about mostly our homeless populations and, underserved populations. And this will mean a lot of times our poorer populations. And unfortunately, the way that our nation works, those poor populations are pretty much programmed by our society to stay poor. So we see that spread of things like AIDS and tuberculosis and other diseases that are often related to immunocompromise, associated with lifestyle choices and living conditions of folks that will be in poor situations or homeless altogether. So you will see that, especially if you guys are spending time at some of our clinics or our hospitals that are in those areas. Like I used to work at Integris Southwest, which did serve a lot of these underserved kind of poorer communities, where we actually saw a lot of tuberculosis. So just be aware of that. It's here in the United States. It's not something obscure. So you might already be aware of that. So that waxy coating, those are our Mycobacterium. The other ones that we have are the ones that have no cell wall. Come on. And these no cell wall, they just literally don't have any cell wall. So it's hard for them to hold their shape. So they don't hold their shape. So their cell shape, instead of being defined as coccus or bacilli, they have another term that we will use called pleomorphic. So morph means shape, and pleo means many. So we have a lot of different shapes that could be going on. These guys often have like random shapes. They can even sometimes have a sort of cockeye appearance or a bacilli appearance, or they might be donut shaped. It's all just one species, like within that one species. These guys all fall within a certain genera as well. So if we're talking about the ones that have the wax, That's mycobacterium. If we're talking about the ones that don't have a cell wall, these are called mycoplasmas. This isn't something like obscure or anything. Mycoplasma pneumoniae is the cause of walking pneumonia. So if you have pneumonia, so we have fluid building up in the sacs of your lungs, but you're still able to essentially basically function. You're not going completely cyanotic. You're not turning completely blue, and you're not bedridden because you're not getting enough oxygen and all this. You're able to otherwise function in society okay, but you do have fluid building up in your lungs as a result of this infection. That is what we call atypical pneumonia or walking pneumonia. And it's most of the time caused by mycoplasma pneumoniae. These bacteria don't have a cell wall. And the way that I remember that is that I think carrying around that cup of plasma, like I said in lecture, if you spill out a literal cup of plasma, if somebody's plasma from their blood, the liquid part of the blood, it's basically essentially akin to spilling a cup of water. It's not going to hold any defined shape. These guys don't hold a shape because they are pleomorphic, because they don't have a cell wall. Okay. So that's how I remember the difference between mycoplasma and mycobacterium. All right, moving on, the Gram stain. It's important to understand how the Gram stain works. We're absolutely going to be going over this in lab 3, which happens on our review day. So it's either Monday or Tuesday for you guys of week 4. And so we will be doing that in the lab before the exam comes. So we'll have some actual hands-on practice seeing it and how it works and functions and everything. But let's just really review this quickly. If I'm explaining to you guys about how we have this scaffolding built up from those sugar chains held together by those peptides, well, when we put in crystal violet stain, our first stain, that crystal violet gets kind of trapped in this scaffolding. All right. So it's really thick like that. If we just had one layer, and let's just assume this box is just an example of that. Okay, let's say that's just one layer or very, very few layers of peptidoglycan. This guy will kind of hang out in there, but it doesn't really get trapped by all the chemical bonds and everything like that. So we'll put the stain on. So for our gram-positive guys, these guys are going to get stuck in that thick layer. But for a gram-negative, it's just going to get kind of put there temporarily. Next thing we do is add iodine. And iodine, what it does is it's something called a mordant, like brick mortar, that helps fix things into place. So what our iodine does is it sort of fixes these guys all in place so they can't get out of that trap that they're in. But because the crystal violet in a gram-negative is so loosely trapped in there, that iodine can't trap it. Iodine can't fix it. It can't fix crystal violet in place in our gram-negative. Okay. So your cells are still going to look purple here because the crystal violet hasn't gotten rinsed away yet. But we're in the process of fixing things that can get fixed in place, fixing them in place with the iodine. So that's what that step is all about. Our next step, though, is going to be the alcohol. And our alcohol is our decolorizer. So any crystal violet that didn't get fixed in place by the iodine will get rinsed away. So that means this crystal violet would have gotten rinsed away. So we'll rinse that away in our gram-negative. And that's because it didn't get fixed in place by the iodine, literally. So when we rinse with our alcohol, our gram-positives, all that purple that we put in there, that's going to stay there, even when we rinse with the alcohol. So our gram-positives are going to stay purple. Okay. Our gram-negatives are going to rinse clear when we add the alcohol. So our decolorizer literally will decolorize at this point. And then we will add a counter stain. And that's just so we can see our negative cells, because otherwise they'd be colorless. It's almost impossible to see colorless cells. So we add the red in so we can have some contrast between our positive and our negative cells and still see our negative cells. So we'll add safranin as our contrast color. When we add safranin into our gram-positives, that purple is such a strong color, you won't really see that red peeping through. So that's why gram-positives are purple and gram-negatives are red. All right, so that's how that will work. Next, we talked about endospores. with endospores, endospores are our protective, completely dormant structures that certain bacterial genera will form to be protected from the harsh conditions of their environment. These structures are not metabolically active. They are not reproducing. They're not doing anything. So if I were to ask you a question on an exam and I say, you step on a nail and you get an endospore into your flesh through that puncture wound, and later you get tetanus. Was it the endospore that produced the toxin leading to your tetanus, or was it the vegetative that made the toxin that led to your tetanus? It was the vegetative. The endospore cannot make toxin. It's not making anything. Is dormant. It has to become vegetative again. That's the active, metabolically active, reproducing, dividing version of the cell that can do things. The version of the cell that does things at all is the vegetative. The endospore is completely inert, does nothing. So it has to revert back to that vegetative state in order to do anything. It's very important that we remember that as well, because endospores ain't going to hurt you. It's when they turn back to the vegetative version that you start having illness. So all of the work is being done by vegetative cells always. Vegetative is also a term that we can use for any bacteria that are doing anything metabolically active, even if they can't make an endospore. So Staphylococcus aureus, it can't make endospores. In fact, the only genera that make endospores are either Bacillus or Clostridium. So that'll be in their name. And so Bacillus anthracis, the cause of anthrax, makes endospores. Clostridium difficile, the cause of C. diff, that makes endospores. Okay. Staphylococcus aureus is not Bacillus and it is not C. diff. It's not in the name. Excuse me, Clostridium. It's not in the name. So if it's not Clostridium or Bacillus, it doesn't make an endospore. If it is Clostridium or Bacillus, it makes endospores every time. Staphylococcus aureus doesn't make an endospore, so it's always vegetative. But Bacillus and Clostridium can switch between if it may need to avoid those really rough times. As it turns out, Clostridium species hate being exposed to oxygen. It'll kill them. So if there's oxygen and they sense that there's oxygen, they will form endospores to protect themselves until they get in a better sort of situation. All right, that's endospores. There's a whole life cycle involved with the endospores. Be aware of the terminology involved in those. Remember, endospore is the dormant structure. Vegetative is the active multiplying version. And when you are forming an endospore, you become a sporangium. You're the cell that is forming the endospore. You're a sporangium. And you only make one endospore. One cell to one endospore. That's it. This is not a reproductive method. Okay. So when you're an endospore, you have this nice thick cortex to protect you. And then if you want to go back to being a vegetative cell, you will go through something called germination. And that's just the process of going back to the vegetative. That only takes about an hour and a half. Pretty quick. So anyways, that's the endospores. And then we sorted out all of our eukaryotes based on their cell envelope. So real quick, I'm going to talk about that. Just to summarize that, we have 4 groups that you can fall into with basing off of just that cell envelope structure. Okay. So our 4 groups for the cell envelope structure characterization grouping. Cell envelope groups. The first one, I always start with firmicutes. Firm because they have a thick cell wall. Okay, so our firmicutes, which ones have the thick cell wall? The ones that trap the purple, right? So that's our gram positives. The next ones I'm always thinking of, well, if that's our gram positives, where do our traditional gram negatives fall? And that would be the one that has the next gra, the word that has gra in it, the gracillicutes. The next one I always remember is the Mendoza cutes. Remember how I thought Joey Mendoza was cute? And that was a long time ago. So that's archaic. So I think of archaea being from a very long time ago. Its term is what essentially what that means. So archaea, a long time ago, I thought Joey Mendoza was cute. So Mendoza, cute. So that's archaea. And if that doesn't work for you, one other thing you can remember is that we have another group of cells that could fall into the sorting hat, if you will. And this is the tenericutes. Tener, think of that as being tender without a D. So this is literally like this. And then imagine that it didn't have the D, that would be tener. But tender, because they don't have no cell wall. They don't have no cell wall. They are cell wall-less. So this one is going to be our mycoplasma. That's where our mycoplasmas will fall. And so those are our groups for how we're going to basically group our bacteria and archaea, all of our eukaryotes, based on their cell envelope structure. And that's the summary for chapter 4. So I'm going to go ahead and move on to the missed topics. So for our missed topics for this week, for chapter 4, we're going to start first with gram-positive versus gram-negative. We took some time to basically show the differences there. If I am drawing this out, then, you know, if our gram-positives would look like nice, big old thick layer of peptidoglycan. And what's really going on in here are these multiple levels of these sugars that are actually, you know, cross-linked essentially with those peptide chains, right? So this is what's really going on in here. If I were to take the time to kind of give a representation of what the gram-positive looks like. Okay, so our gram-negatives, again, just real quick, gram-negative is going to be maybe just this. So we might have a chain, a sugar chain, and a sugar chain, and our gram-negatives might just have that one kind of layer going on. Stop. All right. So now you can see when we're actually doing the staining procedure, how when we put crystal violet in here, this is all going to get stuck in here. And it's easy for iodine to come along and make that get fixed in place. Hold all of that in place in that big meshwork, that scaffolding of that peptidoglycan. Whereas whenever we add crystal violet to the gram-negative guys, it'll kind of stain it and stay there associated with it because of the charge of the cell. But what happens is whenever we rinse with our alcohol, we are actually rinsing away all of that purple. It's not going to get fixed in place by the iodine like it did with our gram-positives. So gram-positives will stay purple. The gram-negatives rinse clear when we get to the alcohol step. And so they're going to be clear. It's going to be like impossible to see them. So we add saffronin as our counter stain, and that is going to stain our gram-negatives red. And our gram-positives are going to stay that violet from that crystal violet, so they are purple. P positive, purple. Red to me is a negative color. That's how I remember the difference, okay? So that's what's really going on. With our gram-positives, they also have an extra molecule in the surface of their cells that just kind of helps maintain the structure of this very thick multi-layer scaffolding. And these molecules are called lipoteichoic and teichoic acid. So we have that on here somewhere, I believe. Yeah, here we are. So our gram-positive guys have that to help maintain that. Our gram-negatives. So keep in mind what's actually going on with all of this. This is just the cell wall portion of your envelope. What's really going on is you will have your cytoplasmic membrane. Then you will have your cell wall. Made of peptidoglycan. That might be thick or it might be thin. And then only gram-negatives will have a outer membrane. That's to give a little bit more structure and support, as well as control of kind of what's getting to that inner level of that cytoplasmic membrane, because they don't have that thick cell wall to support their structure and the shape of their cells and all of that. It's not going to protect them as much. They just have that one thin little layer of it. That extra outer membrane, still also just a lipid bilayer, is going to give that extra support. So that's what's going on structurally as far as the cellular envelope of these guys. So because it's the thick layer of that peptidoglycan, the tecoic and lipotecoic acids, those help out with the structure of that for the gram-positives. The gram-negatives don't need that because it's not very thick. So what a gram-negative will have, gram-negatives have on their outer membrane, because they need that outer membrane for extra support and shape and control, they have something called lipopolysaccharide. Lipo, here we're talking about fatty acid chains. So these are hydrophobic regions. This is what sticks them into the actual membrane itself. Polysaccharide, we have many sugars attached to this, and that LPS, that lipopolysaccharide, and the sugars are polar, which we talked about in lecture. So LPS is sugar fatty acid chain thing on the surface of that gram-negative outer membrane. Okay. Another word for LPS is endotoxin. It's the same thing. Okay. And this is something that our body automatically recognizes as foreign and will start initiating the inflammatory response to, including causing fever, and it can eventually lead to development of shock and can be deadly for you. So great parents like new students. Why would great parents like new students? Because, hey, we're great parents. We can't wait for our kids to learn and whatever. So gram-positive. And so those are going to have like our lipotecoic acid and things like that. And then our negatives are going to have outer sugar. I mean, it's just one way to kind of help remember with however you guys want to make your own. You don't have to use my stupid mnemonics that I'm offering you guys for these things. But yeah, so positive for peptidoglycan. That's how I kind of remember it. You have a whole lot of peptidoglycan going on with our positives. Positive, P, purple, positive. You have thick peptidoglycan. Negatives, red is a negative color. Also, toxins are negative, so they have endotoxins. So there are other ways that you can remember kind of what's going on with all of that. Let's move to our example questions. The patient sample is tested and you find lipotecoic acid. So what is lipotecoic acid before we even get to our choices? Remember, lipotecoic and just tecoic acids help maintain thick walls of gram-positives. So we're talking about gram-positive organisms here. So we can say, if we hear that it has tecoic or lipotecoic acid, we're talking about gram-positives. And then what's our question? What can you infer about the sample? Well, I'm going to probably be looking for gram-positive here. And as it turns out, that's one of our choices. So we know if there's tecoic acid or lipotecoic acid, it's gram-positive, period. It can't be gram-negative. They don't have tecoic acid. They don't need it. They don't have the thick wall that would require the maintenance. Acid fast bacteria don't really need the techoic acid. You don't need to know this, but they just happen to not need that because they have the waxes that are supporting everything in their walls, but they do stain gram positive. But gram positive blankets, pretty much all of our gram positives will have the techoic and lipotechoic acid. Defining feature of our acid fast is going to be that mycolic acid. Lay down, buddy. Meatball. Lay down. You cute boy. Yeah, you're cute, but lay down. So we can tell that this is going to be gram positive. So this is not our answer either, right? So we know why that's our right answer is gram positive. Next question. Which of the following is not a feature of the gram positive outer membrane? Uh-oh. So you should be looking at this question and being like, excuse me, what? Because gram positives have a thick cell wall, but they do not have an outer membrane. Okay. So you should be reading that question and being like, uh-oh, Professor Yost done set us up, but I'm not going to fool you, right? So which of the following is not a feature of the gram positive outer membrane? No, none of these, because we don't have a gram positive outer membrane. Lipotechoic acid. This is found in the cell wall of gram positives. The cell wall. Lipopolysaccharide. This is the outer membrane. This is an outer membrane of gram negatives. Peptidoglycan. This is found in all bacteria walls. So it's D. All right, next topic. This is going to be discussing capsules. Looks like, yes, this is capsules. Capsules are a densely associated protective layer surrounding bacterial cells. So it's densely associated. It's made mostly of sugars. It's sticky. It's for adhesion. It's for creating biofilms. And it's helpful for evading phagocytosis. This page goes into a little bit more detail on this, a little bit more elaboration on this. So if you're having a hard time understanding the purpose, the function, and the structure of the capsule, this does talk about that a little more. I'm not going to go over it necessarily in a lot of detail. We do consider a capsule to be a virulence factor, which is a topic that we'll talk about more and more as we go through the semester. But what a virulence factor is anything that will enhance pathogenicity, this next part down here. Pathogenicity is the ability to cause disease. And what is disease? You guys remember what it is? Disease is deviation from perceived health. So the ability of a microbe to cause deviation from perceived health. Anything that helps a microbe achieve that is a virulence factor. So obviously, if it's helping avoid being eaten up by your white blood cells, which is phagocytosis, that's helping that organism cause disease. All right. Evading the immune response. So you can have slime layers, which are very closely related to capsules. They're just loosely associated. And then we have very well-defined capsules. These are the typical ones that we're going to be talking about in the course. Just a firmly attached, denser version of it. So protection, adherence, virulence. These are functions that or effects of the having a capsule. Capsules prove by all vital clues. It's just reminding you guys, little mnemonic here. Vital clues, basically talking about the virulence factor aspect of these capsules on our bacteria And there is breakdown of these in my notes on some of these guys. I didn't make one on this one in particular, but some of my mnemonics, I have explanations for them. How do I hide this? There we go. Oh, that's not what I wanted to do. Cool. All right. So for our questions for this one, a bacterial strain has been genetically modified to produce a capsule with altered polysaccharide composition. So this is a nice fancy way of basically saying different sugars than usual in our capsule. So what's it going to tell us about that? Researchers observe that the modified bacteria show reduced virulence. So this is a fancy way of saying that our change in our capsule made them less effective at causing disease. So different sugars in our capsule decrease disease. So we can define it that way. Based on your understanding of capsules, so this is our actual question. Now, which of the following best explains the reduction in virulence? So it's asking us which of the choices would explain why we don't cause the disease as badly. Do we think it's because the altered capsule composition, the different sugars, are preventing the bacteria from adhering to host tissues? We know capsules are important for adhering, so it could be this one. This one's possible. So I'm going to put a star next to it. The modification, the changed sugars, enhances its ability to evade phagocytosis. If we're enhancing our ability to evade our immune cells, that would be increasing virulence, increasing disease, not decreasing. So it's not this one. Does the altered capsule, does the different sugars disrupt bacterial cell wall? That has nothing, the capsule has nothing to do with the cell wall. Just because we're changing the capsule, which is an extra sugary coating, just like being coated in honey, is that going to have anything to do with the cell wall? This has nothing to do with peptidoglycan. The capsule is a whole different structure. So it's not going to be this specifically because of that. And then the modification inhibits, so the changing of the sugars, does that inhibit the synthesis of exotoxins by bacteria? We didn't talk much about exotoxins really at all, but an exotoxin, it's in the name. And if you guys come up to a term that's on the test that we didn't discuss and you don't know what it is by looking at the term, you can always raise your hand, call me over, and ask a question about it. Worst case scenario, I might say something like, well, that's a term that you should have learned during the lecture. This is not one. We didn't talk about exotoxins yet. So this particular situation is going to be a situation where if you asked about this, I'd be like, an exotoxin is a toxin that bacteria secrete outside of their body. That's why it's called exo. It'll be outside of their little cell. Okay. So these are secreted toxins. So does a change in that sugar coating on our bacteria inhibit our bacteria from secreting our toxins? We did not mention that. We have not talked about that being an issue with our capsules. In fact, our capsules, we talked about being with adhesion, evading phagocytosis, and with making biofilms. None of that talks about affecting, enhancing, or having anything to do with secretion of our toxins. So we're going to rule that out. We definitely did talk about adhering being a property of it. And you can imagine that if you're taking your honey coating that you have on you and changing with the sugars that are in your honey coating, that could make it less sticky and less able to adhere to surfaces. Adhering to tissues within your host is an important way that our pathogens infect our body. So our answer is A. Our next question. In a laboratory experiment, researchers treat a bacterial culture with a chemical that degrades polysaccharides. So these are sugar chains. So we have a chemical that breaks down sugars like what we have in our capsules. All right. After treatment, that bacteria's ability to evade phagocytosis, a function of our immune cells, is impaired. So the chemical breaks down the sugars in the capsules, and now bacteria can't outrun or evade or escape. Say escape. Escape our white blood cells, our immune system. Okay. That's what's happening. What conclusion can be drawn about the role of the bacterial capsule in this context? So we kind of already have an idea of the three major roles of the capsule: adhesion, evading phagocytosis, biofilm. This is clearly, if we are breaking down the sugars, that the capsule would is now being unable to help the bacteria from evading, getting away from our white blood cells eating them up. So let's go through our choices and see what we have. A, the capsule polysaccharide component is crucial for evading phagocytosis, while the protein component is not involved. So this is a possible choice. We didn't talk much about the protein component, but they can have proteins involved in the actual capsules. If we're degrading the sugars, clearly that sugar portion would be involved in that sticky adhering aspect, as well as, which we talked about in the first question, as well as our ability to sneak away from our white blood cells. So this is a possible answer. Second choice, the protein component of the capsule is essential for evading phagocytosis, and the polysaccharide component does not contribute. We're breaking down the sugars, and now we can't evade. It's the sugars. So it's not the protein. Both polysaccharides and proteins in the capsule are equally important for evading phagocytosis. Now, this one's a little trickier. We can't really rule this one out necessarily, but we know that the sugars are important for evading phagocytosis. So A is the one that we're leaning more as the most correct in the choices so far compared to C. The chemical treatment suggests that the capsule does not play a role in the bacteria's ability to evade phagocytosis. Clearly it does, right? So we can rule that out. So A is our breakdown one. Again, the questions on the test shouldn't be quite this intensive. They'll have some critical thinking, but it shouldn't be quite this intensive. But if you can think through why this is the right answer here, then we're definitely in good shape. All right. Chapter 4 for endospores. We talked about endospores being completely dormant and inert. That's not what I meant to do. Why are you not letting me? There it goes. They do nothing except wait. For better conditions, okay? So these guys, germination is the step where we're going from endospore into vegetative. Vegetative, aka active metabolic, dividing, reproducing. All right, so that's germination. Sporulation is going from vegetative into sporangium, leading to the formation of an endospore. A sporangium is just the cell making the endospore. That's what it's called, okay? So this is the process basically that's involved in all of that and the terms that are going to be relating to that. The species that are able to do this will be called Bacillus something or Clostridium something, okay? So Bacillus and Clostridium, as it tends, as I guess just this is a rule of thumb here, all endospore forming bacteria and therefore all Bacillus and all Clostridium are gram-positive. Okay. It just helps having that thick peptidoglycan layer, helping with creating our thick coating and everything. Just think of it that way. So for endospore, ENDOSPORE. Ever, never dies. Organisms survive perilous, only reluctantly emerging. They're only reluctantly going to emerge if they have the thing that's going to wake them up, usually a water or a nutrient, to cause them to revert back to their vegetative or active, making toxins, growing, reproducing, all of that stuff. Okay. Otherwise, these endospores can essentially live forever, even in very harsh conditions, including heat, radiation, freezing, all sorts of stuff. Moving on to our questions. A researcher discovers a bacterial strain that forms endospores. Okay, so we know it's a bacterial strain that forms endospores. So automatically, we also know it's either Bacillus or Clostridium. Okay, we know that. We know it can switch between endospore and vegetative. We know the endospore is dormant, the vegetative is active, and we also know that they're gram-positive automatically. All right, so that's what we know automatically. But this particular strain lacks genes that are associated with its coat. So it's protective coat. So it's going to have a less protected layer. Okay. That's what it's telling us. After exposure to extreme heat, which normally endospores can withstand, turns out this strain has reduced survival. So it's a less protective coat. It's susceptible, or we'll say sensitive. Because it's less protective, it's now sensitive to heat. What conclusions can we draw from this observation? Is this strain due to a lack of DNA replication in the endospores? Okay. Is it not surviving as well because of its DNA replicating? Is it because the spore coat impacts its ability to withstand environmental conditions? I mean, that's pretty much what it's telling us in the question, right? So this is probably our answer. I want to read through our other choices though. Reduction, because that's the problem that y'all have, right? Not maybe you guys specifically, but we know that many students have this issue. Please read all of your choices before you finalize your choice. The reduction in survival rate suggests that the bacterial strains endospores have a higher metabolic activity. This is not suggesting anything about metabolic activity. It's also unlikely that the DNA is being impacted in any way, but we do know that we are being having gene changes that have led to being sensitive to environmental factors like heat. The strains endospores are likely to be more resistant to chemicals. More resistant? That's not what I'm getting out of this at all. We're actually saying it's going to be probably, if we were to put chemicals on it, I'm betting that it's also going to be more sensitive to that as well. So B is our answer. All right, our next question. A patient of yours is diagnosed with C. diff, Clostridium difficile. Why would containment protocol be more strict for C. diff than for Salmonella? So we already know endospores, your Bacillus or your Clostridium. So does Salmonella have endospores? It does not. We know that automatically because it's not called Bacillus or Clostridium. Compared to Clostridium difficile, which does have endospores. So why would you need greater containment for C. diff? Well, it's not because Salmonella has endospores. That's ridiculous. Our next question. C. diff is endospore forming. Okay, now we're talking. Gram-negative bacteria all form endospores. It's actually gram-positive. This isn't even talking about the gram stain of these guys, but all endospore forming bacteria are actually gram-positive. All diarrhea-causing bacteria are endospore forming. Clearly not, because we know Salmonella causes a diarrhea. We know that other organisms can cause diarrhea, and they don't all form endospores because they're not all called bacillus or clostridium. So this is not our answer. So this simple statement of Clostridium difficile being endospore forming is our correct answer. But obviously, if you need to protect from these endospores, which can survive chemicals, they can survive heat, they can survive all of these harsh conditions, then clearly you're going to have to practice being careful not to spread those endospores around, because it's going to be hard to clean that up. They won't dry out, nothing. So you want to protect from spreading those around. All right, next we have our cell envelope classifications. These were these 4 groups that I kind of went through earlier, how I remember them. This is their little breakdown: firm, grace, mature, tender, sort of similar to kind of how I was kind of talking about how I remember it, but you remember how you like to do. This was just one other suggestion of FGMT: firm, grace, mature, tender. Firm, thick cell wall. Gracie means thin skinned, so they have a thin cell wall. Okay. Mature, long, older, archaic, archaea, and then tender, tenere, which actually kind of means tender skin in Latin. So that's how we would be categorizing our eukaryotes based on their cell envelopes. Ripley, quit. She's like wallering, as my husband says, just rolling and rubbing all around right on my, like, next to me. Calm it down, my girl. So let's answer some questions regarding this way of grouping our bacteria. They're all 3 right here by me. I'm in bed still. Only Spaghetti's really being a nice, calm boy. All right. Ripley. No. You're very needy right now. Microbiologist is studying a bacterial strain that has a thick peptidoglycan. All right. So why, if it has thick peptidoglycan, what do we know about it? It's gram-positive. It's got that thick wall, which they're literally just telling us that, right? Thick wall. It's probably got, because it's got a thick wall, it's probably firm. So we can probably already guess where this is going. Which of the following groups does it fall into? Is it the firmicutes? Yeah, it's the firmicutes, firm and cute, right? It's not our gracilicutes or our mendozicutes or our tenericutes. It's a lot of words. It's really just asking you, where do our thick peptidoglycan layer guys fall in these 4 groups? Here's a whole lot more words. A 56-year-old patient presents to the hospital with severe GI symptoms, okay, including nausea, vomiting, and abdominal pain. Tests reveal that the causative agent is gram-positive. So if it's gram-positive, again, we know it has a thick wall, a peptidoglycan. It happens to be a rod. Which another term for that might be bacillus. That's describing the shape of the cell, right? That doesn't mean that its name has to be bacillus. Other organisms that might not have its name be bacillus can still have the bacillus shape. But if its name is bacillus, then we're talking endospores, right? But this guy happens to be gram-positive. It is rod forming. It is rod shaped, excuse me. So it is bacillus shape. Upon further testing, we see that the cell wall is thick. That's what gram-positive means. And it shows resistance to desiccation and high temperature. It's very resistant to those things. The physician suspects that it might be due to a common bacteria in the soil that can form endospores. Well, that would explain the resistance, right? Capable of surviving extreme conditions. Based on these details, which of the following bacterial groups is most likely responsible for the patient's symptoms? This is a whole lot of information in a whole lot of words that is just asking you, where would you group a gram-positive organism? Firmicutes, right? Thick cell wall, firmicutes, Gracilicutes, that's our gram negatives. Mendozacutes, that's our archaea. And Tenericutes, these do not have a cell wall. All right. So that is it for our review for chapter 4. I took a little time to go over a little more explanation of these since they are pretty key points for upcoming stuff. But that is it for today. Does anybody have any questions for me before we go? All right. So if you guys don't have any questions for me, yep, I'm happy to help. Then I'm going to go ahead and call it a day. If you guys think of any, of course, always email me, and I will have this video and the summary posted later today, probably. And of course, let me know if you guys have any questions. And on Monday and Tuesday, we will do chapter 5 for the eukaryotes. On Wednesday and Thursday, we will go over the viruses. And then on the following Monday and Tuesday of week 4, we will do our review where I will give my own presentations over 8 individual presentations over my 8 topics, which is how I'd expect you guys to treat it in the future. And I'll try to save some time to go over those critical thinking questions so that we can talk about that a little bit as well and how to work those out so that you're a little bit prepared for those types of questions on the exam. Otherwise, I bid you adieu, and I will see you guys next week. And have a good weekend.